Thank you very much for the very kind words and for reminding me of all of the places I've been to. And thank you to the organizers for the invitation and for organizing this conference. I mean, one of the things that actually you'll understand relates to my talk is how impressive it is to see a conference like this one. When I started in the field 20 years ago, I don't think we would be able to have an ending age-related diseases conference with so many people. And really the excitement and all of the activity, both academia, industry, even at government level now in aging is very exciting. So, well, I guess for a conference on aging and ending age-related diseases, I don't need to remind you of the importance of tackling aging. So instead, I'll just show you a cartoon. I guess the point that we're all convinced of is that we need to tackle aging. We need to develop interventions that allows us to at least retard or delay the process of aging, that allows us to reduce or even eliminate age-related diseases, which are now the major burdens of medicine and of society. And if we could do that, if we could develop one way, as I think in the previous discussion, was if we could at least develop one way of retarding aging, now that would have huge medical, health, social, and of course financial benefits for whoever was able to do that. Now, just a couple of slides of introduction on in terms of where we are in terms of manipulations of aging. So we now know that the process of aging is plastic, and that means that we can manipulate aging in model systems, in animals, by genetics and by interventions. We can knock out individual genes, and this has considerable effects in the level of aging, both accelerated aging and retarding it. So I think this has really been a revolution in the field, and one of the reasons why there's so excitement about the field of aging now. So one of the things I actually started when I was a PhD student many years ago was a database called the GeneAge Database of Aging-Related Genes. I'm sure many of you are familiar with it. But what I think is quite impressive is how much we have advanced in our understanding of the genetic basis of aging. So we now know of over 2,000 genes in model systems, so in mice, flies, worms, and yeast, that when individually manipulated have a significant impact on aging or longevity. So I think this is remarkable. I mean, so in worms alone, you can knock out a single gene, or it's an H1 allele to be more specific, and you increase lifespan by 10-fold. And in mice, you can knock out a single gene, I think one of the growth hormone receptors, and you can extend lifespan by up to 50%, as well as retard a variety of age-related diseases. And again, I think this is a remarkable breakthrough in the field. I'm convinced that some of the pioneers, like Cynthia Kenyon, Tom Johnson, will receive the Nobel Prize for these advances at the genetics of aging. And that's one of the reasons why really there's been all of this excitement about the field of aging that we see reflected here in this conference. Likewise, there's a lot of advances in terms of pharmacological interventions in aging. So another database we have is the Drug Age Database of Age-Related Drugs. We've actually just updated 2 days ago, just before I came here. We now know of over 500 drugs or compounds that in mortal organisms significantly extend lifespan. Now, the effect sizes of drugs and compounds are not as great as those you see from genetic manipulations, but it's still very remarkable how much we've advanced in pharmacological manipulations of aging. And again, it's one of the reasons for all the excitement in the field. And so it's not surprising that we now want to harness all of these advances at the genetic and pharmacological level to develop interventions, to go into the clinic. And so we have this growing and exciting anti-aging biotech sector. And we've heard for some of these companies, we've heard about Calico already today, probably the most famous company in this sector, although, well, as Vera pointed out, quite secretive, I would say. Unity and Senolytics and many other companies. I mean, this is my disclaimer as well of companies I'm a consultant or advisor for. So really the point is that there's been a lot of advances in model systems to understand how we can manipulate aging. And not surprisingly, there's a lot of academic labs and companies trying to capitalize on that. So trying to harness what we've discovered to develop human therapies. Which again, if we could make that work, would be tremendous, which would be revolutionary, as been pointed out before. Now, what I wanted to focus, however, today is but what if we fail? So the question I posed myself before I came here, and that I want to pose to you, is what are the possible reasons for us to fail? If 20 years from now, nothing of this have happened, we have actually not achieved in developing interventions based on what we know of the aging, what are the possible reasons for that? And I was thinking about that, and I came up with a few possible reasons. Maybe there's others you can think of, and if so, I'm happy to discuss afterwards. But my idea for this exercise is, okay, if we can think of reasons for why we will fail or why we may fail, then maybe there are ways then of mitigating these possible reasons for us to fail. So I'll give you a couple of reasons. Those I thought would be more obvious, or those I could think of. Again, maybe I'm missing something. And then I'll give some ideas as well as how we can mitigate risks based on these things that could go wrong. So one of the first reasons I think we could fail is, well, maybe what we know about aging is wrong. Now this is the Hallmarks, very famous Hallmarks of Aging, which I'm sure most people in the audience are familiar with. But I mean, I remember when I started in the field, probably the oxidative stress, the oxidative damage, the free radical theory of aging was the most popular theory of aging for why we age. And you know, that's changed. I mean, part of it because there was a number of findings from model systems, in particular mice, showing that, well, failing to support the theory. But the point is, a lot of this is based on recent research that could change in the next 5 or 10 years. The reasons for why we think we age may ultimately be wrong. There may be something else to it, but something may be something more specific to it, but ultimately we may not, and given the complexity of aging, we may not be looking at aging from the right angle. And I think that's an hypothesis we have to accept, and I think it was Vadim Gladyshev in the debate that pointed this out. As a possibility of how we don't fully understand the process of aging. So that is something to consider. Maybe what we know about aging is wrong. Now, the other possible reason for failure would be that, well, we know a lot about aging in animal models, but maybe this does not reflect what happens in human beings. Maybe we human beings, maybe the hallmarks of aging are correct, maybe the reasons for the causes of aging we think are important in animal models, but perhaps they're not in humans. And there's reason to think that may be the case, actually. Some evolutionary biologists have pointed out that, well, we know a lot about manipulations of aging in short-lived animal models. But of course, maybe what makes a mouse live longer, or makes a short-lived animal live longer, well, that's not going to work in humans because we're already long-lived. And so perhaps some of the longevity drugs, genes, and pathways that we're discovering in animal models, they work very well in animals, but they're not going to work in humans because we're long-lived already. So as an example, for instance, I would go back to cell senescence. We've heard, I mean, there's a lot of excitement about senolytics, about cell senescence. I'm sure you're familiar with the papers from the Mayo Clinic showing that ablation of senescent cells leads to improvement in mice during aging. So I do think there is evidence that cell senescence or senescent cells contribute to degeneration during aging in mice. But we actually know very little about the role of cell senescence in human aging. We know there has markers of cell senescence been identified in human tissues, particularly in certain pathological conditions, but whether that's causal or not, we still don't know. So the role of cell senescence in human aging, I would say, remains unknown. And I mean, for more details, I would point out a review I published with João Pedro from the Mayo Clinic last year that goes into more detail on the reasons why I argue that we don't know the role of senescent cells in human aging. So this is another possibility, is that we actually know at least some of the causes of aging in animal models, but they don't apply to human beings. So the other possible reason for us to fail is something that's been touched upon, is limitations in, or how long it takes to do clinical trials in humans, which I'm sure has been touched upon already today in this conference. You know, there's also regulatory hurdles in terms of doing experiments, not experiments, in doing clinical trials for aging. And whoops, I went. So, and the other problem is that specifically in aging, because we don't have biomarkers of aging yet, we do have long validation times. I mean, as been pointed out today already, even an experiment in mice to test a particular compound or therapy will take a certain amount of time, which is going to be quite a long compared to other fields like cancer research. So that is a major limitation. What it means is that we can only test a limited number of therapies for a limited number of conditions. And so maybe what we pick to test, well, it turns out to be wrong. Maybe it's the wrong choice, and we test something that doesn't work, or we test it for a condition that doesn't work, because we can only test a certain amount of therapies, or drugs, for example. So we might have been the role where we test all of these things, and there's a lot of excitement about it, but they all fail. That is a possibility. So the question then is, all right, so that's not very optimistic, is it? But how can we improve our odds? Now, one of them is through technological advances. I mean, I show here a photo. This is me pretending to work on a DNA sequencing machine. The point is that I'm sure you're aware that there's been lots of advances in DNA sequencing, and that means, I would say, in omics technologies in general, our ability to generate data has increased a lot. Like other technologies, we're hoping advance. Like stem cells and regenerative medicine and gene therapy, because our field depends on other technological advances, as it happens in science. I mean, if you wanted to build a mobile phone 100 years ago, I mean, you could put all the money in the world into it, you would not be able to do it, because the technologies were not there, and you would not be able to develop those in sufficient time. So our field is also dependent on other advances in technologies. So how can we improve our odds? Things like omics technologies, I think, help. And this relates also, how can money accelerate progress? So what are the best ways of reducing the risks from an investment perspective and from a perspective of the whole field? So one of the things that's been touched upon already today, and I briefly touched upon it, is in terms of biomarkers. I do think that would have a tremendous positive impact if we could have suitable biomarkers of aging to reduce the amount of time it takes to do experiments and to do trials. So, for example, without going into a lot of detail, that's something, I mean, my lab is working mostly at the level of gene expression, which is not as accurate biomarkers of epigenetic signatures, but I find them more interesting because they tend to be more functionally informative of what's going on. For example, we've done a lot in terms of gene expression signatures of aging, also a caloric restriction. I mean, we've recently started working on, excuse me, on gene expression signatures of cellular senescence as well. So we actually have a bioarchive paper. I don't have time to go into details. We have a paper in bioarchive showing that some of the gene expression signatures you see in cell senescence is also observed in aged human tissues. Again, it doesn't prove that cell senescence is important or is causal, but at least we see some signs of it at the gene expression level. So related to this, I would also argue that given all of the advances in technology in generating data, particularly at the omics level, we also need better approaches to make sense of the data. And again, that's something we've been trying to do in employing data-driven approaches, for example, to prioritize genes and drugs for experimental validation. I mean, the goal, going back to the issue that it takes time to do experiments, is that we need to reduce the number of experiments. Something has been touched upon by some of the speakers today already. Well, we cannot test everything. So if we can prioritize and identify compounds and genes for experiments, that gives us a higher chance of success, then that would advance the field. And as a very quick example, I mean, this is something we published already, and I mentioned at previous talks, but I mean, a couple of years ago we did a project on repurposing drugs for longevity. So we employed gene expression signatures of longevity, specific caloric restriction, and then we looked at the overlap with drugs using the connectivity map that Vadim Gladyshev mentioned already, and then we identified a number of drugs. I mean, one of them was actually rilmendidine, which he discovered as well. And then we tested 5 drugs. 4 of them extended lifespan in worms. I guess the most interesting of them was allantoin, which had not been previously related to aging, which is actually interesting, a marker of oxidative stress, and was whose function we're now studying. But, I mean, we've published this a couple of years ago. The point is that we can employ this network pharmacology approaches to identify and prioritize compounds for experiments so that we can reduce the amount of experiments we are doing. I mean, the last thing I would argue that is helpful is to incorporate human data. I mean, that's something, I mean, we have another database of human longevity genetic variants. And the other thing we're doing is we're using public data sets like the Framingham Heart Study and UK Biobank to associate genes with aging phenotypes, which we then try to link into what we're working on in model systems. And likewise, we can use existing human data, for example, to see if some of the compounds that we're interested in have any sort of effect in patients. So as a specific example, we're working with Tim Spector from King's College in London. So basically, to investigate some of the drugs we identified or we tested in worms, some of those drugs that we're trying to reposition, they're already applied, they're already used in the clinic. So we're seeing if in the patients that they have, there's any sort of benefit from taking those drugs compared to other drugs. For example, at the level of Horvath's epigenetic clock. So we try to use this human data to, again, try to infer what are the most promising, or at least the drugs and genes that have the highest probability of being relevant to humans. Now, having said that, I do think there is one problem we do have, which is at the level of how long it takes to do experiments. We can reduce the amount of times it takes experiments, but how long it takes is not something we can easily change. So, you know, whether you're working with mice or you're doing clinical trials, you know, you still have to take that amount of time to do those experiments. And it doesn't matter how much money you have or how much resource. You might be able to do more experiments, but that amount of time to gather that data, that is not something that is easy to change. And I do think that's a major bottleneck that we have in aging research that we cannot easily change, even if we have all the resources in the world. There is one possibility I'll just briefly mention of how this could change, which is if we could actually reverse aging. If we could have an intervention, I mean, by me, reversing aging means reversing, you know, age-dependent mortality. So I see mortality as the major outcome of aging. So if you cannot reverse mortality, you're not reversing aging. So if we could actually develop a way of reversing the process of aging, now yes, that would again be a game changer, even from a testing perspective, because it would have a major impact. So that is something that would, in theory, really change things. I don't see, I mean, it's not possible to reverse aging in model systems so far. So I don't think it's something that will happen in the foreseeable future, but if we could somehow do it, then that would really be a game changer. And this is something related to the question I asked the previous speaker of, you know, should we go really for something that is high risk, but that will really change, that would be really changing the whole field. So reversing age would be pretty amazing. I mean, that's actually a photo of me from about 20 years ago when I started in the field. Yes, I did think my hair back then looked very cool. So in summary, we do know that there's a lot of excitement about all of the genes and drugs that can modulate longevity in model organisms. I think that, I mean, this has been fascinating and it's been incredible. It's really generated a lot of buzz about the field, some of the previous speakers has mentioned. Having said that, I do think it's important that we try to anticipate failure, we try to predict why we could fail, so we can then minimize risks. At the level of, okay, maybe we don't understand aging all that well, particularly human aging. As I mentioned before, I mean, humans are not just big mice or gigantic worms. I mean, there are differences between species. Again, something previous speakers have touched upon. So what works in mice does not necessarily work in humans. I mean, I don't know, I think we've cured cancer in mice hundreds of times, but it doesn't necessarily mean we've cured cancer in humans. So what works in mice does not necessarily work in humans. And again, we have this very long experimental time in our field in particular of aging, which I think is a big bottleneck. I think is a big issue that is not easy to change, no matter how much technology or money we put into it. Having said that, there are ways, I hope I persuade you that, you know, employing data-driven methods and integrating human data of trying to minimize risk and trying to improve the odds that what we're discovering from model systems will work in human beings. And so with that, I mean, they really didn't speak much about the work in my lab, but we do some work in my lab as well. And these are the people who do the work in my lab, and these are the funders that paid for the work in my lab. And if you're interested in the work in my lab, please feel free to visit our website and drop me an email. Thank you very much for your attention.